Hello everyone, this is Mr. Schultz, and this is AP Biology video 1.3. This video is going to cover nucleic acids. This is the Biology in Focus section, Chapter 3, Section 6. So, nucleic acids. Nucleic acids, their main role is to code for something. They're basically the genetic code of our cells. And they code for what's called a gene. A gene is a specific section of DNA that codes for a very specific protein or a macromolecule. There are two types of nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Their differences are the sugars in their backbone, and we'll get into that a little bit more in a minute. The whole role of nucleic acid is, yeah, it's genes, but what does that mean? Well, we have our DNA, and our cells are able to make a photocopy of it. We call that mRNA. So you're going to notice like DNA to RNA. And that RNA is then going to move to a ribosome where it can be read as instructions, which can then make and pull in amino acids to make a polypeptide chain, which will eventually fold and make a protein. Now, proteins are really important because they do almost all the work in our bodies. And so in order to get all of those proteins together, we need to take the DNA, the instructions, and we need to turn them into instructions the cell can use and then make the actual product itself. So there are three parts to a nucleic acid, or the parts of the nucleic acid. So nucleic acids are made up of a bunch of monomers called nucleotides. The nucleotide has a nitrogen-containing base that's going to give it the letter in DNA, like A, T, C, and G. It also has a 5-carbon sugar. In DNA, it's deoxyribose, so the sugar is deoxyribose, and in RNA, it's a ribose. And they have slightly different structures, which causes them to have slightly different shapes as well. And they generally have one or more phosphates as well. They actually start off with 2 to 3 phosphates before they're synthesized together, and those phosphates get pulled off, and that's how they get put together into their other polymers. A polynucleotide is going to be either DNA or RNA. So we take a whole bunch of the nucleotide monomers and we put them together to make a polymer. In DNA and RNA, there are the nitrogen-containing bases. And there are the pyrimidines, which is cytosine, thymine, found only in DNA, and uracil, found only in RNA. And they're here. And you can tell their structure because they have only one ring. Purines, which are adenine and guanine, have 2 rings. And so if you're asked for pyrimidines and purines, the C's, T's, and U's are pyrimidines, and the purines are adenine and guanine. The odds of them asking you like which one's a purine or pyrimidine on the AP Bio test are pretty slim now. It's kind of one of those rote memorization things, but it's useful to know that there are differences. It's also important to know which letters go with which. And so in nucleotide polymers, the nucleotides are attached by what's called phosphodiester linkages. So these are the bonds that form. And it results in a backbone of a sugar phosphate, and then what's going to happen is another sugar and phosphate, and it just keeps doing this over and over again. And so each end is different. And we call them 5' and 3' depending on the carbons you're looking at in the ring of the sugar. And in between the backbone, so if you look at this model here, here's the backbone, the phosphates and the sugars. In between them is where the nitrogen bases are. And they don't actually link up like this. They don't like lock together that way. There's actually going to be a hydrogen bonds that form between them. And what's really interesting is because there's 4 letters, and the letters can be in any order and can be repeated and done in all sorts of crazy combinations, there's an infinite possibility of letter combinations, which means that there can technically be an infinite number of different kinds of proteins that could be assembled. It just depends on which proteins have evolved for whichever functions are needed. The polymerization reaction is really interesting. So it's kind of like an ATP molecule with the 3 phosphates. And what happens is the 2 phosphates at the end break off along with the hydrogen here, and it forms the phosphodiester linkage. And it'll just keep doing this. And it does it in a very specific order. It's always 5' to 3'. If you're confused as to why, it's kind of like when you pet a cat. If you pet it the right way from head to tail, the cat's happy and purrs. If you try to go from tail to head, the cat's not happy and things don't work right. And so our cell is always 5' to 3' when assembling both DNA and RNA. So, DNA structure. DNA consists of 2 strands forming a double helix. A double helix is like a spiral staircase. The backbones run in opposite directions. We call this anti-parallel. And so one is going 5' to 3' while the other is going 3' to 5'. And so depending on which way you're going, the DNA is read in opposite directions. The backbone is on the outside while the bases are kept on the inside of the DNA. And the strands are held together by hydrogen bonds between the bases. And so if you pay attention, if you look at and pay attention to what I'm talking about, A's and T's go together and they form 2 hydrogen bonds, and C's and G's go together and they form 3 hydrogen bonds. And these bonds are what kind of help hold the DNA structure together. So A's always bond with T's and C's always combine with G's. And so we call these complementary strands. So if you have like A, T, C, G, you have T, A, G, C. And so that's what we mean by a complementary strand. And both strands can carry protein instructions for proteins, but they might not necessarily line up in the same way. RNA is a little different. So DNA is double helix, and so there are 2 strands of DNA stuck together. RNA can't do that. And so it forms a single strand. But that's not to say that RNA can't clump together. In fact, its nucleotides do form hydrogen bonds within the actual mRNA molecule. So DNA kind of lines up like this. RNA can kind of fold in on itself. And this actually allows RNA to perform certain functions within the cell. An example of this is like rRNA, ribosomal RNA, is able to hold itself together to be part of the ribosome, or tRNA, which carries amino acids to the ribosomes. They're actually folded in on themselves. They have a really cool kind of loopy structure. One thing to note is that in RNA, A's pair up with U's or uracil instead. There is no thiamine in RNA. So when we look at RNA, you just got to remember that the T's are U's instead. And then RNA tends to be much more variable than DNA does. So if you have a virus that's got RNA in it, and like a retrovirus, they're much more likely to mutate and change because their structure is less stable than a DNA molecule. So just keep that in mind when you're looking between RNA and DNA. So DNA viruses tend to be a little more unstable, which means they can mutate a little easier than a DNA-based virus does. One thing to note, though, and this is really important, is that when we look at DNA, it's a major, major piece of evidence for evolution. So shared traits in organisms provide evidence of shared history. This means that a shared trait is a shared gene, so the DNA is the same, or really, really similar. So shared DNA can also show shared ancestry. So we can look at like certain markers within our DNA, certain sections or genes, and we can compare them to other people, and we can use that to help us determine if we're related to somebody or not. So when we're looking at DNA, we share more DNA similarities within our family than other people. So like if you have siblings, your DNA will be much more similar to your siblings than it will be to like the random stranger that lives across the street. But humans, in the whole population of humans, they share a lot of their DNA with each other than we do with, say, a chimpanzee or a mouse. We still share a lot of DNA with these species, but it's just that within the human population, the whole population on the planet, our DNA is really similar to each other. And when we look at it, we can also look at like, oh, well, we share more DNA sequences and similar sequences with chimpanzees than we do with horses or with fruit flies. Or we can even compare ourselves to plants. So this nice little 23andMe image shows like, hey, this is approximately how much DNA we share with these other species. And if we look at our DNA, like we share about 44% of our DNA with fruit flies, which seems weird. We are very different from fruit flies, but a lot of our cell structures are really similar to each other. And so like the codes for the proteins that get things in and out of the cells might be really similar. They might be a little different because the functions are a little different, but they're similar in their structure, so their DNA is similar as well. So when we look at DNA, we can use it as a piece of evidence for evolution. The more DNA you share, the more sequences that are similar, the more closely related you are to that organism or group of organisms. So this concludes the nucleic acid PowerPoint, and we'll be covering proteins in our next video.